It's now time to introduce the second inaugural lecture of the day, of the evening, and that will be presented by Professor Selina Wray. So Selina is a professor of molecular neuroscience and continuing with the tradition set by Tamarin, Alzheimer Research UK Senior Research Fellow. And she is in the Department of Neurodegenerative Disease at the Institute. Selina received her degree in biochemistry and biological chemistry from the University of Nottingham some time ago, before undertaking her PhD training in Diane Henger's lab at the Institute of Psychiatry at King's. Her PhD was awarded in 2008, and Selina then joined the lab of Professor John Hardy at the Institute of Neurology as an Alzheimer Research UK Junior Research Fellow. During this time, Selina spent time as a visiting researcher in the group of Thilo Kuhne at the University of Edinburgh, and Rick Livesey at the University of Cambridge, before being awarded a senior research fellowship in 2017. Selina's work has pioneered the use of UCL of human-induced pluripotent stem cell and organoids, bringing this technology into the fabric of the building and working closely with both clinical colleagues and basic scientists. And she continued to focus on genetic form of dementia, understanding with them the basis of sporadic Alzheimer's disease and frontotemporal dementia. Her work was recognized by several awards, and including the 2018 AI UK David Haig Early Career Investigator of the Year Award, and in 2014, the Red Magazine Woman of the Year Award in the Pioneer category. With the occasion and without further ado, I would like to congratulate Selina for her new position of Deputy Director of the Institute in Communication and Partnership. Thank you, Selina, for giving your time for this important and nerve-wracking function in the Institute. And personally, I was also lucky to interact with Selina while we were writing the UK Dementia Research Institute proposal and spending few hours discussing experiment and how to set this institute up. And more recently, in fundraising for the new institute. So, Selina, thank you for being with us and for presenting your inaugural lecture tonight.
Thank you so much, Gippy, for the kind introduction. I will just share my screen. Hopefully you can all see and hear me. I mean, first of all, just to start by saying what a huge privilege it is to have the opportunity to give an inaugural lecture. It was one of the nicest bits of news that I received during lockdown, and I still can't quite believe it's true. So hopefully this will make it feel a bit more real. I wanted to start by just saying what a huge privilege it is to share the evening with Tam. Tam's a good friend of mine, and I really think of her as being almost a scientific sister. We got our junior fellowships at the same time, our senior fellowships at the same time, our proleptic appointments on the same day, and now our professorships at the same time. So it really feels fitting that we should share this occasion, and I only wish it could be in person. Beyond Tam, I feel privileged to be part of a cohort of people who I can really call friends as well as colleagues. And I didn't have a photo of everyone, but I just wanted to share this of Ed and Sonia. This was just after we got our promotions and we decided it was time to maybe brush up on a few of the kind of background facts that might be useful for our job. And then finally, although I will talk about them a lot throughout the course of this evening, I really wanted to thank my lab. They have always been really supportive of me. I think a professor is only as good as the lab makes them. And so I really want to thank them for their hard work, particularly through the pandemic. And finally, on this slide, I think just to say I would also like to thank them for keeping me grounded. And in amongst all of the excitement, they did buy me a gift from my professorship to remind me that I'm only just okay. And this is, if you see me taking a sip of water, this is what I'll be drinking out of tonight. So where did it start for me? Well, it really started in a town in South Yorkshire called Barnsley, which is about 200 miles north of here. You can see here the kind of coat of arms for the town. Most people, when they think of Barnsley, will think of coal mining or glass blowing, which were the two main industries of the town. And so it might not be, science might not be the obvious choice as a career, but when I was at secondary school in Holgate School, which you can see in this picture here, I really developed a love of science subjects. And here is the first photographic evidence, I think, of me in a lab. This was in our GCSE chemistry lab. There I am at the back, age 15, and I really have my love of science. I really have to thank a lot of the amazing teachers I had along the way, particularly Mr. Summers, who's shown here. He was our chemistry and physics teacher and really taught the subject in such an engaging way that I couldn't help but be interested enough to want to take it further. Outside of science, I think I also should kind of pay tribute to the first places where I really learned the meaning of hard work. And before and throughout university, I had two jobs in Barnsley. One was at the Barnsley Trades Club behind the bar and as a glass collector. And the other was at William Hill in Barnsley Town Centre. And science is hard. No one would deny that science is a hard job, but I really think I've never worked as hard as I have when I've worked Grand National Day at William Hill and then gone on to do a full Saturday night bar shift. It was really here that I learned the meaning of what hard work really is. They also really set me on my path, although they might not seem obviously connected to science. It was on my last day at work at the Barnsley Trades, the kind of collection from the customers gave me the money to buy the textbooks that I needed at university. And also one of my colleagues at Barnsley Trades who drove me and my mum and all of my stuff to Nottingham University because we didn't have a car growing up. And so again, I really look back fondly on the time I spent in both of these places. But it was at the University of Nottingham where I really started to develop my scientific interests. And I spent my 4 years there mainly in this building, the Queen's Medical Centre, where we're co-located with the medical school. And my personal tutor throughout this time was Professor John Mayer, who you can see in this photo here. And his own research was focused on the ubiquitin proteasome system and how it becomes dysfunctional in neurodegeneration. And I remember sitting in his office for tutorials. He would have a framed photo of a Lewy body on the wall, and it really just piqued my interest into how protein misfolding can really be so critical for neuronal health and neuronal degeneration. The other thing that they really did in Nottingham was to encourage us to try and get hands-on experience as much as possible, because learning science is quite different from doing science, as many of us will know. And I'm really grateful to Rob Layfield, who gave me my first taste of research. And that was in the form of a Wellcome Trust vacation scholarship. So it was a paid summer internship. I wouldn't have been able to do it without that funding. And I worked on a project with Rob looking for substrates of the E3 ubiquitin ligase parkin. And to do this, we were really trying to pull down interactors of parkin from pig brains. So I used to, every Monday morning, leave my bucket of PBS in a designated room in the Queen's Medical Centre. I would come back 4 hours or so later, and someone had taken the bucket, been to the abattoir, and come back with some penny dropped. But at some point, walking around the corridor of the university with my bucket of pig brains, I clearly thought, yeah, this is the life for me. It's the, you know, it's the glamour of the job that's so appealing. Joking aside, I think Rob, you know, I'm forever grateful for the time and attention he invested in me. And he really gave me a full experience of what research is like, both working in his lab, but also the kind of enjoyment that you could get from scientific meetings. And he brought me to a ubiquitin proteasome meeting held by the Biochemical Society here in London. It was the first time I'd been to London, the first time I presented a poster, and it really helped me decide that research was something that I would like to pursue. And so when I was working with Rob, I was having a look for PhD projects, and having sworn that I never really wanted to move to London, I found the ideal PhD project. So I thought, well, you know, I'll just move there for 3 years for my PhD. And that took me to the Institute of Psychiatry at King's College London and to Diane Hanna's lab. And again, I think before I talk about the science, I want to just kind of spend a moment talking about the research culture. I think we really hear a lot these days about how difficult a PhD is and how toxic lab environments can be. And my experience couldn't have been further from that. Diane and her group were so supportive, and the wider department in general, so the groups of Jean-Marc Gallo, Chris Miller, and Brian Anderton at the time, really made it a fun place to work and to do science. And I hope my experience at the Institute really set the tone for what I want my own lab to be like. These were some of my lab mates at the time. I'm still in touch with many of them, which is a real pleasure to still have them as friends. Many of them are listening in today. And I'm sure several of them are known to people in the audience, particularly anyone working on tau. So Wendy Noble, who I still chat to multiple times per week, and the same with Amy Pooler, generally saying, you know, does this email look okay before I send it? And I'm really grateful for their kind of continued mentorship throughout my career. So it was at the Institute where I really became fascinated with this protein called tau, which has been a theme in my research ever since. Now, tau is a microtubule binding protein. It binds to and stabilizes the microtubules within neurons. And what that means is it really is responsible for giving the neuron its structure and its transport system. Now, what I find fascinating about tau is it's an incredibly diverse protein. There is diversity in its splicing. So there are 6 different tau isoforms, and we know that the exact expression of those isoforms is really important both for health, for disease, and for development. And like many proteins, it's also subjected to really extensive post-translational modification. And the most commonly studied is its phosphorylation. So phosphorylation is really essential for the regulation of tau's normal function. Without that phosphorylation, tau can't bind to the microtubules. But we also know that phosphorylation is a key modification in disease. So you see this hyperphosphorylated tau, which form these aggregated clumps inside the brain cells, particularly neurons, but sometimes glia. And this one protein is found in the pathology of many, many diseases. And these are really clinically heterogeneous. And I find this fascinating. How can this one protein be a central player in diseases that have completely different symptoms? And it's a question that continues to fascinate me to this day. But the approach we took when I was working with Diane was to really look at post-mortem tissue and ask, well, is the biochemical signature of tau in these different diseases different? And the answer is yes. This was my first ever paper published from my PhD. You can see some of my Western blots on the side here, where with these different diseases, hopefully what you can see is that the tau has a different barcode that will characterize that disease. We combine this with mass spectrometry to look at the phosphorylation of tau in the different diseases. But perhaps the most exciting finding was this fragment that you can see here, which really is present only in a subset of the neuropathies. And I'm really proud that the work on this fragment continues in Diane's lab today. Most of the brain tissue that I used in the project actually came from Queen Square. And I remember Diane bringing me as a very nervous junior PhD student to meet Tamas and Janice. And I really clearly remember on the train into town, Diane saying it's good for you to get to know people outside of our institute, and it's especially good for you to get to know people at the Institute of Neurology, because one day maybe you will want to work there. So yeah, that was a prophecy of Diane's that really, really came true. So before I move away from the Institute of Psychiatry, I just want again to kind of pay tribute to some of the people who I worked with there. As I say, many of them are friends to this day. We used to call ourselves Team Tau. You can see hopefully from the pictures that it was a lot of fun. I'm absolutely sure that this is not comprehensive, but everyone here has been, incredibly supportive as an informal and formal mentor. They also really encouraged me to send what I didn't realize at the time would turn out to be probably the most important email of my career. And when I was finishing my PhD, it coincided with John moving back from the US to set up his lab at Queen Square. And I remember I was looking for postdocs and everyone saying to me, this is a perfect opportunity. John will be setting up his lab, he will be recruiting, you should kind of email him. And at the time, I was maybe not the most confident PhD student. I still felt kind of a little bit nervous, but I remember crafting this kind of very carefully worded email, running it past 100 people until I think it must have been Wendy who said, just send the bloody thing. Just nothing bad is going to happen. And I sent it at 4 o'clock on a Friday, saying, dear John, I've heard you're moving back to London. I'd really love to come and work with you. And it was this probably one of the best emails I've put together. And within 5 minutes, I got a reply from John saying, sounds good, come and see me on Monday, which is a very typical John email, but which, as I wouldn't be sat here if things hadn't worked out. I came and met him in the Brunswick for a coffee. We had a really lovely chat, and I moved to his lab 6 months later. John has been probably one of the most important mentors during my career, and I think it's worth spending a minute or two just to kind of give thanks for that. For this audience, I don't need to talk about John's scientific merits. We all know he's a great scientist and has made a lot of the fundamental breakthroughs in the field. But for me, John has he has a big brain, but an even bigger heart. He's such a kind and thoughtful mentor. I remember, having not been here for too long, him saying, I always try to do the best by everyone, and that doesn't mean that I do the same for everyone. And that really stuck with me in the way that he kind of thinks about people's career development and doesn't try to think of a kind of one-size-fits-all formula. He is, I am really grateful for everything he's done and all the time that he's invested in me. And it's been kind of a lot of fun working with him. When I moved to John's lab, it was also the start of what turned out to be one of the other long-term important professional relationships in my life, which was with Alzheimer's Research UK. So I got a fellowship not long after moving to John's lab, which was from Alzheimer's Research Trust at the time, and then my senior fellowship with the rebranded Alzheimer's Research UK. And actually these images were both taken at Alzheimer's Research UK events, but 10 years apart. So it really has been kind of a long-term supportive relationship. And, I think, as Tam said, they're doing really great things for dementia research, but also for raising the profile of dementia as a whole. And I'll talk about them a bit more later. So when I met John and kind of talked about what I could do here, I have to say I was really panicking before, because it was the era of the genome-wide association study, and I know absolutely nothing about genetics. So I came to meet him thinking, oh God, he's going to quiz me on genetics and I won't know a thing. But actually the conversation took a different turn, because it was at the same time that induced pluripotent stem cell technology had just been developed. And this is the ability to take an easily accessible patient cell type, such as a fibroblast or a skin cell, induce the property of pluripotency. And what that means is that the cell is a blank slate. So we can give it the correct signals in the lab to differentiate into any cell type of interest, and that includes neurons. So the reason that this is so important, of course, is because the brain, the human brain, is really inaccessible to us during life. And this has been revolutionary for our understanding of neurological disease. And I hope it will bear through into the development of better treatments, because again, people in the field will know we've cured Alzheimer's in a mouse many, many times, and it's not always translated to human benefit. So this is really another tool in our toolkit. And what he said to me when I sat down with him is, you know, this technology is going to change the field, but also there's no better place than Queen Square to do it, because where else do we have such well-defined patient cohorts, particularly of monogenic diseases, where we can take their cells and develop a dementia in a dish model? And so that's what I came here to do. It was one of the other reasons that I wanted to come here, because I really believe in the power of having preclinical and clinical science together. And I think we're really fortunate at Queen Square to have clinical researchers who are excited about and want to collaborate with the lab-based science. And so we set this up initially with Martin Rosser, who was hugely supportive and really keen for us to get the ethics so we could do this from patients at the Dementia Research Centre. And then later on, obviously, I continue to collaborate really closely with Nick, who's been another amazing mentor through my career. And again, I think as well as really wanting to understand the preclinical science, wanting us to understand the clinical science and what the patients face. He's also been a fabulous mentor in other ways, particularly in making sure that as a junior researcher, I would always have a seat at the table. And Gippy mentioned in his introduction how we spent time writing the DRI application together. I think that's kind of one example where, Nick would say, we need to have, the people who might be driving this and doing the work to sit together and really put this application. So I'm really thankful for everything he's done to champion me and to kind of lift my profile within the Institute. I should also thank, as we talk about the cells that we've collected, Susie Barker, who is one of those rare people who I couldn't find a photo for on Google, despite my best efforts, but she's really been instrumental in helping us to get ethical permission to do this study, which is a long process, and I couldn't imagine tackling that without Susie's expertise. But we eventually got it, and for a number of years now, we've been collecting fibroblasts or skin cells from individuals who come to the dementia clinics here. And these are people who know that they have a genetically defined dementia. We ask them to donate a piece of skin so that we can make their stem cells and their neurons. And over the years, I've been really humbled by the patient response to this. Nearly everyone says yes. And I think that's a testament to the DRC and to the other clinicians here, particularly Henry Holden as well, who really invest the care so that patients are excited to participate in research projects such as the ones we do. The collection of the fibroblasts has been a real close collaboration with several friends and colleagues. I have to really thank Natalie and Colin, who took the first skin samples from Alzheimer's disease and FTD respectively, and really quickly turned into good friends of mine. In this photo, we're in the Guinness factory in Dublin. Colin now sadly is over in Australia, so I think it's about 3 a.m. there, so I know he's going to watch on playback, but I saw that Natalie is online. So yeah, I'm so grateful for their friendship and their support. And then of course, John Rohrer, who runs the FTD empire, and we've been really lucky to collaborate with him on a number of projects and have this sustained support of our sample collection. These are some of the papers we published with our dementia IPS. I won't have time to go into all of them, but very briefly, I will outline some of our work on familial Alzheimer's disease and also on tau. Before then, a little diversion. I think when we entered into this, although Queen Square is the natural home for this work because of our rich patient resource, at the time it was not a natural home for stem cell work. We didn't have the full range of expertise that we needed to establish this. And so I took a little detour as a visiting researcher up at the University of Edinburgh. It has a rich history in developmental biology, most famously Dolly the sheep, who you can see here in the Museum of Edinburgh. But I was so fortunate to move up there at a time when they were moving into their new building. This is the Scottish Centre for Regenerative Medicine, which was really a place where I learned what an ideal cell culture facility would be like. My first day was the annual away day. So this was my first day in Edinburgh, on the top of Ben Lomond, which we had beautiful weather. I actually got sunburnt, which you would not anticipate in Scotland, perhaps. And the person who we moved there to work with is in this photo here, Thilo Kunath, who was working on Parkinson's disease at the time with Mike Devine, but was so generous to host us in his lab so that we could learn how to make stem cells from the skin cells that we have. And these are our first stem cells, the first ones that I made. They're from an FTD patient with the tau mutation. You can see the images of the lovely stem cells here and also their nice chromosomes. And I can't, words can't do justice to how much better my IPS work is for spending time in an environment where the focus is developmental biology. I think really having the deep knowledge that Thilo has on kind of pluripotency, the mechanisms governing it, the quality control that you need to do to work with these cells has really informed my mindset working with the lab. And I'm so grateful for him and for his lab, and also other friends I made there, including James, who I think is listening tonight, who really kept me company in many, many hours of cell culture. And we continue to be in touch to this day. Once we have the stem cells, we need to know what to do with them. How can we make the cell type that we're interested in? So before I returned to UCL, I spent a little bit of time working in Rick Lives's lab in Cambridge, where they had developed a great method to make cortical neurons from induced pluripotent stem cells. And you can really capture the process of cortical development in a dish, which I think is really exciting. And the cells look beautiful, as you can see here. They look like a firework that's exploded, but also they don't just look like a neuron, they behave like a neuron. And that's what you can see in this image here. I'm really grateful to the people in his lab who took the time to kind of show me the cell culture, particularly Peter, who's in this image here, Natalie and James, who this was the only group photo I could find, was of the Gurdon Halloween party. So you just have to trust me that they're in there somewhere. There were also two other things that happened during my time in Cambridge, which were really exciting as a scientist. The first one is that as we made this video, I came back to UCL and we hosted a lab visit via Alzheimer's Research UK for the Health Secretary at the time. And this happened just before the G7 summit on dementia. And they were so kind of enchanted by this video that it ended up on the big screen with the Prime Minister and lots of kind of government ministers in the audience. So it was really nice to see what sort of an impact that engagement can have. The second thing that happened is while I was in Cambridge, John Gurdon won his Nobel Prize, and he's still very active in the lab, and he brought his medal in for us all to see. So this is, here I am holding a genuine Nobel Prize medal, which is the closest that I will get. So that was a real exciting thing as a scientist. So following Cambridge, I came back to Queen Square to kind of set up this technique here. Back to Queen Square, but with a new location. So I moved out of Queen Square House and into Wakefield Street, which is where I am at the moment. And we were really fortunate that through John and through Tom Warner, we had two cell culture labs specifically dedicated for IPS work. And that's needed because the cell cultures are very long-term, very susceptible to infection. And we really needed that extra capacity. So it's through the refurbishment of these 2 labs funded by the Brain Research Trust that we're able to do what we do now. And the other great thing about Wakefield Street is the great culture that we have here. I think it's really a fun place to work. People are kind, generous, and we do some really nice things. This was at a public engagement event. This was at the Oval of one of our kind of annual outings for cricket trips. And I'm really grateful to people like Rena and to Rohan for making the also gave me a chance to recruit new people, and Ellie and Charlie were my first 2 recruits as a lab. Ellie is a PhD student and Charlie is the first postdoc I recruited. And as I've said at the beginning, we're only as good as the people who kind of make us. And I'm so grateful that these were my first 2 recruits because they really made the lab what it is. They're good fun, they get on well most of the time. This was a bit of a kind of jokey fight at a picnic we had, but I'm also really grateful for the creativity that they bring, both in the lab, whether that's as a kind of new PI when we had to improvise on some kind of forms of lab equipment that we didn't have, and also outside of the lab through the invention of our lab cocktail. And this is at one of our Christmas parties where Charlie invented the Jaeger bell, which is a baby bell hollowed out with some Jägermeister inside. And I'm showing you this so that you don't feel too sad that there isn't a drinks reception afterwards, because it means that you've escaped trying this delight. But joking aside, again, I'm really grateful because I think they've set the standard for the lab and the ethos of the lab, that when we recruited new people, they kind of become part of that. So what do we do as a lab? Well, as I mentioned, my primary interest was tau, and one of the kind of challenges of tau is its developmental regulation. And so thanks to Teresa, who joined the lab not long after Charlie and Ellie, we were really able to show that the cells we grow in the dish reflect the natural development of neurons that we would expect to see in vivo. And you can see this kind of shift in tau splicing that happens over time and happens over a long time. And we were one of the first groups to really pioneer these long-term cultures, but also to show that if we did this from FTD patients, we see disrupted splicing similar to what happens in the brain. And this set us up for a lot of exciting work and collaborations. We obtained a NC3R's Crackit Award to really develop these human cultures for drug screening and for connectivity studies. That allowed me to hire Agiro Alatsa, who's been a fantastic addition to the lab, works across multiple projects and really keeps the lab afloat in many ways. And it was also a great collaboration with the University of Strathclyde. You can see in this picture Michaeli, Trevor and Graham from the University of Strathclyde, who really specialised in these microfluidic cultures. And these are 2 populations of our human neurons that are separated by this channel that we can then use to understand whether they can connect as efficiently, and whether if we, for example, seed tau pathology on one side of the chamber, does it affect the other side of the chamber? In many ways, this was a perfect collaboration because I really looked for any excuse to go and visit Scotland, and they were always very happy to visit us in London and have a little a meeting always followed by a drink in the Euston Tap. And it was really a fun collaboration working with them. We wanted to push the system even further, and as with any new technology, it really quickly evolves and adapts. So we kind of wanted to move into these cerebral organoids, which are 3D tissue-based models, often referred to as mini brains in the press. And to do this, again, Agiro was really closely involved, and it was really led by Chris Lovejoy, who was a PhD student in my lab. He defended during lockdown and is now a postdoc over in Yale. And these are some images from his beautiful cell cultures, where I hope you can see, compared to the 2D cultures, we have a much more spatial organization of the different cell types that are mixed with different colors, and marked with different colors, sorry. So this is a much more physiologically relevant model of disease. And he again was working on tau splicing. We can see that if we really age these cultures, we get these disease-associated tau patterns. These are really long and risky experiments, so really a lot of credit to him for setting it up. And we're able to now probe these for what pathology is present and how well they mimic the process happening in the diseased brain. The cells also set us up for some really nice collaborations. And one of my first international collaborations was with Karen and Jessica, which resulted in this paper here. So what we were doing is looking at this phenomenon of tau propagation, the spread of tau from one brain region to another, which Karen has characterized very well in vivo. And we were able to show that some of those mechanisms were conserved in vitro in our cells. As well as being a fun collaboration, Karen, for a long time, has been an amazing supporter of mine. I was first introduced to her through Wendy Noble, who was Karen's postdoc. And for the last 10 years, she's invested time in me that she's not obliged to do. We don't have kind of a formal line manager relationship, and yet she's always spared the time to give me great advice, scientific and personal. And she was also a fabulous tour guide in New York. I think this was in 2012, maybe somewhere in the Lower East Side. And I'm really happy now that she's a colleague rather than a collaborator now that she's moved to UCL. And she's also broadened my collaborative network by the amazing people who she hires to work in her team. And it's really great that I get to work alongside Steph on some tau projects now at UCL, and also Natura, who's still in Colombia, but really is helping us a lot with a project in the lab looking at the proteasomal turnover of tau. And that brings me to this slide. I think these cells have taken a long time to develop, but they're really now crucial to a lot of projects in the lab, most of which are kind of pre-publication. But I really have to, again, give my thanks to Georgie, who's a fantastic student in the lab working on tau proteostasis, Dale, who's a case student based at UCB, who's been kind of looking at ways that we can manipulate tau proteostasis, and Tiaisha, who's working with Rohan De Silva and with Tamas in the Brain Bank. And I'm really happy that she sits within part of my lab too to work on projects in tau seeding. So moving away from tau, although it was my first love, it's impossible to be here, I think, and to work with John and Nick and not be excited about familial Alzheimer's disease, which is this very rare form of Alzheimer's disease that is inherited in an autosomal dominant way. And here at UCL, we have this amazing cohort that's been built up over multiple generations by Martin, by Nick, by others. And it gives us a real opportunity, I think, to understand the influence of genetics on disease. And you can see here, this is from my colleague Natalie's paper, where in spite of the fact that all of these people carry a monogenic driver of their disease, there's a huge variability in age at onset, also in symptom presentation. And so the other big question in my lab is, can we understand the molecular basis for this heterogeneity? And this has been in part funded by my senior fellowship from Alzheimer's Research UK, but also by the NIH BRC. This has been a close collaboration with Natalie, with Tamarin, and with Lucia, who's based over in Leuven, and is really a gamma secretase expert, and also a long and fruitful collaboration with Henrik Zetterberg. And this is us on a trip to Sweden with our lab and his lab. So what have we found? Well, the work that I'll present over the next few slides has really been led by Charlie, who I've already mentioned, by Jamie Toombs, who was a fantastic PhD student and is now a postdoc with Tara Spires-Jones in Edinburgh, and by Nanette Williamson, who's now at the Imperial DRI. And very quickly, hopefully what you can see here in this kind of reasonably large cohort of cell lines is that the Alzheimer lines look very different to the control lines in terms of their A-beta profile. And this is capturing in a dish what we think is happening in the patient. So it's telling us that in terms of a model, the cells that we're working with are a good one. We wanted to understand this a bit more by taking, advantage of our unique situation where we have a lot of different aspects of studying the patient to kind of get to the bottom of their disease. And collaboratively with Nanette and with Tam in the brain bank, we can show that the A-beta profiles we see from our cells are reflected in the postmortem tissue from the same individuals or individuals with the same mutations. So it's really adding validity to the work that we do. We also got to look at this in matched CSF with Henrik, and again, we can see kind of similar profiles of A-beta in cells and in CSF, which means that they might be a good tool for surrogate biomarker discovery. And one of the most exciting things happening at the moment is this development of plasma biomarkers. And really, in this study from earlier this year, led by Antoinette O'Connor, you can see that there's a really close correlation between the A-beta production in the cells and the A-beta ratios in plasma. And so what this tells us is the models that we're using are a good reflection of what's happening in the individual. To understand a bit more about mechanism. And so we've complemented our patient cell lines with tools that we've made using CRISPR. And again, this work was led by Charlie, but was also a collaboration with Jenny Pocock and Claudio, who is shown in this photo here. This was after they visited the lab from Peru, and they made us all pisco sours on their last day. So that was quite a nice thing to host. And what we were able to do is use CRISPR-Cas9 to create tools that complement our patient cells. So this is from a patient line with a heterozygous mutation. We could use CRISPR to make the homozygous version, the wild type corrected version, and also the presenilin-1 knockout. And this really let us address questions that have been a source of debate in the field for many years, but for the first time, we could address them in human neurons. Specifically, one of the questions we were interested in is, well, are the mutations gain or loss of function? And again, I'm going through this quite quickly, but what we could show is that the mutations in presenilin-1 that cause disease are only affecting the gamma secretase function of presenilin, which you can see here, this really nice dose-dependent effect of the mutation on A-beta processing. In contrast to that, if we look at the chaperone activity of presenilin-1, we can see that that's not affected by mutations in the same way as it is by knockouts. So they're kind of, the mutations that we're looking at are not a simple loss of function. And this is kind of one of the first times we've been able to address this in human neurons. But of course, what we really want to do is use these for drug development. So the reason that I came here and stay here is because having the preclinical and clinical research side by side, I think gives us the best opportunity of closing the loop. So we take a person's cells, we use them to model their disease in a personalized way, and then we screen them for therapies that we can eventually trial within the same patients. And I'm really excited that we recently recruited Rebecca Gabriel to the lab to work on a collaborative project with Paul and Sarah at the DDI to really develop ways to correct those A-beta profiles that I showed you earlier. And so we're excited where that takes us. Finally, everything I've shown you so far has been kind of static measurements. And the other thing that we're really excited in is this collaboration with Henrik again, led by Ross Patterson and Claire Leckie, who's doing the work in the lab, together with Kevin Mills to look at production and clearance by developing labeling methods. And we've got some preliminary data here from those experiments. And I think this will be really transformative once we get those up and running. Okay, so to just move away from my focus on Alzheimer's and on tau, I think one of the difficulties at UCL is feeling a bit like a kid in a candy shop, and there are so many good things to do that it can be hard to kind of stay focused. But some of these diversions have been a lot of fun. And this is a collaboration where Ellie developed cells collected by Henry and Hugh from C9orf72 carriers. And we did this at a time when we didn't know what the gene was. It hadn't been discovered, but we knew that this patient carried that locus. And so we were able to do this and work closely with Adrian, who's a good friend and collaborator. We developed drugs to really attack the pathologies that we could see in these cells. Another long-standing collaboration and friendship has been with Professor Helene Plum-Favreau. This was born out of me being forced to move office down to the 7th floor, but actually I got a really good friendship and a really good scientific collaborator out of this. And Helene's focus is on mitochondrial function. We could support some of our work early on with our patient cells. We've kind of developed this to develop assays and ways to look at mitochondrial function in iPS neurons. And I'm really grateful to Mark and Daniela in Helene's lab for helping us establish these assays in our own lab. And they're now part of Jackie's PhD, and also Anna, who's a student co-supervised with Sonia, to look at the impact of FT genes on this process. And finally, we've also collaborated a lot with Andre to look at mitochondrial function. Again, this is one example looking at VCP where we could see these abnormal mitochondria. It's been a great collaboration involving my friend Marta and also Ricky at the Crick, and we heard about his VCP work a few months ago. Finally, although today I've really talked about monogenic disease, we're looking at how we can take this to the broader late onset versions of disease. And so I'm really happy to have a collaboration again with a good friend of mine, John Schott, to work with this 1946 birth cohort. So people who were born in 1 week of 1946, who were all enrolled into this cradle to grave study. We have really extensive amounts of genotyping and phenotyping data, and we're now in the process of trying to generate their stem cells so we can look at the cell biology of people who are at risk from Alzheimer's and those who are low risk of Alzheimer's. So that kind of finishes my science bit. I wanted to spend my last few minutes talking about other stuff. Before I move on, there are a lot of people who I've not had a chance to thank through kind of collaborations, but who have really made the Institute a great place to work. Mike, who I started here with, and Patrick, kind of 11 years ago, and lots of other people. And as Tam said, I think we'd be nothing without the unsung heroes such as Rosie and Helene and Elizabeth, who don't get the recognition through papers, but really keep everything that we do afloat. So moving away from science, I wanted to have a slide to thank Alzheimer's Research UK. They've been really instrumental in my career developments through their funding, but so much more than that. And I think working with Alzheimer's Research UK has really given me an appreciation for what goes on behind the scenes to let us do what we can do. And working with them on their fundraising, on their sporting events, and on some of their political engagement has really opened my eyes to kind of the breadth of activity. I'm sure this isn't capturing everyone, but I'm really kind of grateful to work with them. And I feel like I've grown up as a scientist as they've grown as a charity and really exploded. In terms of their kind of influence in the dementia research field. I also wanted to thank Stuart and Tara. They've both been chairs of the SAB of Alzheimer's Research UK. Stuart in particular was my external examiner for my PhD and has been a really strong advocate and supporter for me for the last 10 years. And so I'm grateful for the time he's taken to really kind of be a cheerleader. Aside from science, I think the other thing that I like most is talking about science. We've really made an effort as a lab to take it out and get people engaged with what we do. And these are just some events that we did at the Science Museum, which is really a proud moment of mine, having almost 100 people from UCL and from Alzheimer's Research UK showcasing our work to about 5,000 members of the public. And I think this is kind of one of the things that I'm really, really proud of, that we were able to kind of coordinate and put on this event. It's led to further collaborations, particularly with the Created Out of Mind consortium led by Seb. And I was part of a project called Brains in a Dish with Charlie Murphy, with Chris, with Phil Ball, and others, where we've really tried to develop innovative ways to showcase our research. You can see here a neural disco that we did at the South Bank, which was absolutely mad, but really lots of fun. We were really lucky that we took Phil and Charlie's cells Chris grew them in the lab, and then Phil wrote about it in this book. And it's the kind of the first chapter of this book is just about his feelings about us growing his cells in the lab. And it really gives us a different angle to think about what we do. The best thing about the public engagement is I've been able to take it back to where we started. And, as I mentioned earlier, I think it's maybe not the most common route for people from Barnsley to follow a career in science. And I've been invited back to do some really cool things like lead the graduation procession from Barnsley College. They put my name on a lamppost, and not in a wanted criminal sort of way, in a kind of look at this girl done good sort of way. And now working with Charlie and Alison, who you can see in the photo here, we're developing an art exhibition that will open up in Barnsley next year. And I hope we'll kind of get people a bit more engaged with the work we do. And we're tying back to the kind of history of Barnsley in glass blowing. These are some glass neurons made from old glassware, mimicking the calcium imaging that I showed you earlier, which is something that we're kind of super excited to engage people with the work that we do. The reason it's so important for me is because, I still feel really privileged that I've managed to kind of change what my outcome could have been like. It's not ref returnable, but I would argue that it's maybe more prestigious than a Nature paper, where they really talked about, how someone like myself who grew up on benefits could really still fulfill this career dream that I had. And I really think central to what I do now, apart from the science, is to hold the door open for other people. And there are several ways we do this. One of those has been hosting students from this Into Science scheme, which gives people from underprivileged backgrounds a chance to kind of gain that hands-on experience. And I was really happy to do this with my close friend Amanda. And we're here after we just run the marathon together for Dementia Revolution. And I think if even one person feels that they can kind of make that career for themselves, I'll be really proud of that. And of course, my main motivation for this has been my parents, who were always puzzled and confused why I had such a passion for science. But they didn't mind what I did, as long as I could make a life for myself that was better than the kind of start that we had. And, I feel I sleep easy at night knowing that I've done that. But of course, I'm really saddened that neither of them can be here to see it. I know that they would have loved tonight, and I don't want to finish on a sad note, because I know that the best way I can honour them is to take advantage of the opportunities that I've given. So tonight is really for them. And I'll just finish by thanking the rest of my family, particularly Paul, my long-term partner, who's always been incredibly supportive of me. Family from both sides. I didn't have enough space for photos of everyone, but this is my cousin Keely, my cousin Simon, and my grandma, my mum's mum, who I know were all really proud of me tonight, and my aunts and uncles. And then Billy, of course, by audience demand, I had to show a picture of the cat. So here he is, very dapper for my inaugural. And I'll stop there. Thank you so much, everyone, for listening.
Thank you, Selina, for the wonderful inaugural. And now I call Professor John Hardy for the vote of thanks. John.
Selina, first, thanks for those very kind words about me, which you had me blushing here, but thankfully the video was off. Let me just say what a lucky institution I think we are at the Institute of Neurology. I mean, I think when we look for the next generation to carry the Institute of Neurology, to have such complete, well, not just scientists, but people to carry the vision of the Institute through, both with a passion for the research into what causes these diseases, but also with a passion for bringing others into the disease areas and teaching them, and through public engagement, as well as through scientific publications and through fundraising. And by the way, I didn't know half of what you have been up to, Selina. You've been up to a lot of other things that I did not know about. So it's been a real pleasure for me, and I'm sure for all our colleagues at the Institute of Neurology. we should all be very proud of our younger colleagues who are now such wonderful representatives of the Institute of Neurology and neurology research. I remember interviewing you, as you say, in I think it was Starbucks, as you say, and I remember being impressed by your openness and enthusiasm. And really, you know, it was a great hire for me. I'm glad you came. I'm glad you had the courage to, you know, to send that email to me. Yes, I'm really glad you did. I hope you don't think for too long about when you send emails to me now, and you're a little freer. But it's been a real pleasure. It's been a real pleasure having you here at the Institute of Neurology, not just for me, but I'm sure for all the other colleagues whose pictures you showed. Thank you.
Thank you, John.
With the note of John, we concluded today, this wonderful afternoon. I would like to thank Selina and Tamarin for their lecture, John and Tamas for their vote of thanks, and 160 plus attendees that join us tonight. It has been a privilege, and with this, I wish you a very good evening to all of you. Bye-bye.